discussed the structure of the cilium, flagellum, centriole, basal body, centrosome, etc. Now the last topic that is remaining in this chapter is the nucleus, which is the central structure of the cell, which is regulating all the major activities. Almost all the activities of the cell are regulated by the nucleus only. So first of all, it was discovered by an English botanist named as Robert Brown in the year 1831. So it was discovered by English botanist, England, from the England. So it was discovered by English botanist Robert Brown in the year 1831. Nucleus play a major role in the inheritance as well as gene expression. So function is, it plays a major role in two things. First is inheritance and the second is gene expression, or you can also write gene regulation. Now inheritance, that is the process of transmission of characters or traits from one generation to the next, is in the form of chromosomes only, that is the condensed form of the DNA. It is located where? Within the nucleus of the cell. So it plays a major role or important role in the inheritance, that is transmission of characters from parental generation to the offspring's generation. Gene expression or the regulation, that means when certain set of genes has to be expressed. Genes is a basic unit of inheritance, the fundamental unit of inheritance, a segment of DNA, right? So when gene has to express, when gene has to switch on, when gene has to switch off. Ultimately the meaning of switch on and off is when gene has to produce a protein or not. That all is decided by the nucleus only. Now the certain parameters we have to discuss, like shape, size, number, etc. Shape is generally spherical, but oval, bean shaped and lobed nucleus are also reported. So generally spherical, but also in lobed, oval and bean shaped nucleus are also there. Size, it is very small, 0.5 to 2 micrometers, right? Size is 0.5 micrometers to 2 micrometers. Imagine this small size nucleus is holding approximately 2.2 meters long DNA, right? So size is 0.5 micrometers to 2 micrometers. But when the cell is in its division state, that is when it is performing mitosis, size of the cell also increases. Other than that, the nucleus size also increases. But during cell division, cell size and nucleus size also increases. There are certain cells in the plant kingdom as well as animal kingdom which are devoid of the nucleus. And the best example in case of eukaryotic cell we can consider is mammalian erythrocyte, that is RBC, and in case of plant it is sieve tube cells. Sieve tube cells in case of plants and mammalian RBCs are in case of animals like human beings, you can take. They are devoid of nucleus. That means nucleus is absent. Again, if we talk about the fourth point regarding the nucleus, that is the position. So position of the nucleus is generally not fixed. It changes as per the movement of cytoplasm, and we know the cytoplasm movement is called as cyclosis. So position is generally not fixed. Generally not fixed, but moves as and when cytoplasm moves. And we know the movement of the cytoplasm is basically called as cyclosis. Right? Now coming to the internal structure of the nucleus and external also, what all other things that we are going to discuss in the nucleus are nuclear membrane. We will discuss nuclear membrane, that is inner membrane, outer membrane, perinuclear space. Then we will see nucleoplasm. Afterwards we will also see chromatin. Within this chromatin the genetic material is located, which then condenses to form chromosomes. So ultimately in the last we will also see chromosomes. So all these structures we have to discuss in detail. So first of all, nuclear membrane we will discuss. Nuclear membrane. So basically the entire nucleus is covered by two membranous structure. They are called as nuclear membrane or nuclear envelopes. Right? So nuclear membrane is the membrane or covering of nucleus. Two nuclear membranes are there. There is inner nuclear membrane and there is outer nuclear membrane. Both membrane has its own specific role. We all know that if, let us say, this is a schematic diagram, the nucleus does not look like this, but for the sake of understanding I have drawn. From the nuclear membrane we know ER arises, that we have discussed, right? So entire structure of the nucleus is covered with endoplasmic reticulum, SCR and RER, and this outer nuclear membrane also has ribosomes attached. So outer nuclear membrane, what are the two key points of the outer nuclear membrane? First, it is facing the cytoplasm and it has ribosomes attached. So it is facing the cytoplasm, and the second is it has ribosomes attached, and the inner membrane is basically inside, right? So we will draw the nuclear membrane also. Between outer and inner membrane, let us say these are the outer and inner membrane of the nucleus. This gap that is present between the outer and the inner membrane is called as perinuclear space. So space between outer and inner membrane is called as perinuclear space or internuclear space, right? So let us now draw the structure of nuclear membrane and let us understand the functions of nuclear membrane. The one of the most important function of the nuclear membrane is transportation, right? We will see how. And basically the transportation is specific term is given to transportation, which is called as nucleocytoplasmic exchange. That is, the transportation is taking place between the nucleus and the cytoplasm, or vice versa, that is from cytoplasm to the nucleus, right? For transportation, certain channels are required, certain openings are required. Such openings are called as pores, or to be more appropriate, they are called as nuclear pores, which are present in the membrane, right? Let us say this is inner membrane, and within this space the pore complex, that is nuclear pore complex, it is written as nuclear pore complex or annula, is located, right? How it is drawn? This middle structure is basically the area for nuclear pore complex, or it is also written as annula. So within this annula or the nuclear pore structure, it has its wall and the lumen also present, right? So wall, and within this wall there is a lumen also there. So lumen, this region is basically the nuclear pore complex or annula, annula or annulus, A-N-N-U-L-U-S, nuclear pore complex, annula or annulus. Between inner membrane and outer membrane, we have seen the nuclear pore complex is located, and the ring-like structure, this pore is what we call it as the annulus. The size is 800 angstrom. That means the nuclear membrane size we are talking about. So we can consider that the diameter of the nuclear pore, is also 800 angstrom. But this entire 800 angstrom is not involved in the nucleocytoplasmic exchange. That means the entire 800 angstrom area is not available for the transportation. Only this much part is available, and that accounts for 500 angstrom. So 500 angstrom is the active space for transportation, that is available active space for transportation. Now we discussed that outer membrane is facing the cytoplasm and it has ribosomes attached, so they are protecting the outer membrane. Inner membrane is also protected by some other structures which are electron rich proteins only, and it is not a continuous layer, it is a wavy layer which is called as fibrillar lamina. So inner membrane has wavy layer. Wavy layer means something like this. It is not a continuous layer. Waves, that is a wavy layer, is present. This wavy layer is what we call it as fibrillar lamina, and this is made up of electron rich proteins, and the main job of this fibrillar lamina is to support the inner nuclear membrane. So support inner nuclear membrane, right? And this is what we call it as an octagonal shaped pore, and the number of the pore depends upon the type of the cell. It can range from 1000 to 10,000 per nucleus. This is the number of pores available per nucleus of a particular cell, right? The next structure within the nucleus is nucleoplasm. Like each and every cell has its own cytoplasm. So nucleoplasm is nothing but the cytoplasm of the nucleus. Right? So second structure that we are going to study within the nucleus is nucleoplasm. Right? So basically it is a transparent colloidal material. It is transparent colloidal material in the nucleus, right? Basically the main job of the nucleus is to cell, like DNA synthesis, RNA synthesis, like that. So whatever proteins and whatever enzymes that are required for DNA synthesis as well as RNA synthesis are present within the nucleoplasm only. So nucleoplasm composition wise, it is rich in the enzymes required for A. DNA synthesis and B. RNA synthesis. Now there are approximately 8 to 10 enzymes which are involved in the DNA synthesis, and the mechanism by which DNA is synthesized in the cell is called as replication, right? And the major enzyme, we will be writing the name of one major enzyme which are involved for synthesis. That is a must enzyme, DNA polymerase. Like DNA polymerase 1, 2, 3, like that. For RNA synthesis, the mechanism is called as transcription, right? And the main enzyme is RNA polymerase. Second thing, since protein synthesis regulation is also done by the nucleus only, right? And for the protein synthesis, mRNA is required, or all the RNAs are required. That means mRNA, rRNA as well as tRNA, and they are suspended within the nucleoplasm only. So it is also rich in mRNA, that is messenger RNA, rRNA, that is ribosomal RNA, tRNA, that is transfer RNA. Apart from all these enzymes and these RNAs, nucleoplasm, that is a fluid, is rich in inorganic ions like manganese and magnesium. So it is also rich in Mg+2 and Mn+2 ions. They are basically acting as, I already told you, they are activators or cofactors for all these enzymes to act. The pH of the nucleoplasm is slightly alkaline, that is basic pH. It is approximately 7.4 plus minus 0.2. The plus minus variation of 0.2 is acceptable. So pH is slightly alkaline, and that is 7.4 plus minus 0.2. So whatever cell structure and other details, chromatin, everything will be suspended within the nucleoplasm only. Then the third structure that we are going to discuss is nucleolus. Afterward we will discuss chromatin and then chromosomes. So the third structure is nucleolus. Nucleolus is basically a darkly stained body within the nucleus of cell, right? And this term nucleolus was given by Bowman. So term was given by Bowman, right? It is associated with NOR, that is nuclear organizing region, that is basically satellite DNA, associated with NOR region. NOR full form is very important. See, all these things are not given in NCERT, but they are required. Nuclear organizing region. Nuclear organizing region, where in the satellite DNA or the satellite chromosomes. Basically, satellite chromosomes or DNA are the repetitive sequences which are different in all the individuals on this earth, and such sequences are called as VNTR and STRs. That we will be discussing 12th, not now. That is variable number tandem repeats, short tandem repeats. This nucleolus is without any membrane. It is without any membrane. genetic material is localized, and that is very very important structure. Chromatin is basically location. I already told you all the things are suspended within the fluid part of the nucleus, that is nucleoplasm. So it is suspended in the nucleoplasm. Suspended in nucleoplasm. Chromatin is basically a mass of genetic material along with the proteins, basic proteins. So chromatin is defined as it is a mass of genetic material composed of DNA and proteins, and these proteins are histone proteins, or you can also write it is a mass of genetic material plus proteins. Proteins, I already told you, they are histone proteins, right? Normally histone proteins are of 4 different types, that is H2, H1A, H2B, H3 and H4. So these are histone proteins. Histone proteins are rich in basic amino acid like lysine and arginine. So histone proteins are rich in basic AA, that is amino acids, lysine and arginine, and these basic amino acids are providing the positive charge to these amino acids. These positive charged proteins, that are histone proteins, in an octameric form. So histone octamer binds to negatively charged DNA, and DNA is negatively charged because of the sugar phosphate backbone. It is negatively charged. PO4 minus bond is there in the DNA. So histone octamer, which provides a positive charge due to these proteins, amino acids, lysine and arginine, binds to the negatively charged DNA, and that process is called as condensation of DNA. And finally chromatin gets condensed to form something that is called as chromosomes, right? Because the entire length of the DNA is 2.2 meters long. This 2.2 meters long DNA cannot be placed or fitted within the very small size of the nucleus that we have already seen in the initial lecture only, the size of the nucleus, right? Now talking about how this arrangement is done. Basically it is an histone octamer. So 2 molecules of H1A, 2 molecules of H2B, 2 molecules of H3, 2 molecules of H4. 2, 2, 2, 2. That is 8 proteins. So they form an octamer like this. Right? And within this octamer, our DNA, at various locations, these octamer proteins are located. So let us say all these are octamer proteins, and within this octamer protein, the chromatin fibers get attached. It wraps like this. I have to use the, suppose this is the thread of the chromatin that is starting, it wraps like this. And then it continues to form a structure, that is what we call it as a chromosome with its primary constriction. Now the complex that is formed of 2 components, that is histone proteins and the DNA, this complex is called as nucleosome. So what is nucleosome? This is also you have to remember. It is nothing but the complex of histone proteins and negatively charged DNA. Apart from histone proteins, Within the nucleus there are non-histone proteins also. There are non-histone proteins, and the main job of the non-histone proteins is to provide the support, that is structural stability to such structures. Right? Now this gets condensed to form a chromosome. So this is the last thing that we have to discuss, that is chromosomal morphology. Right? So chromosome has this primary construction, that is we call it as centromere. These are the chromatids, the sister chromatids, and surrounding these centromeres there is a protein structure which is called as kinetochores are present, which is mainly involved in the spindle fiber splitting and the spindle fiber formation. Right? Now based on the location of the centromere, there are total 4 types of chromosomes: metacentric, submetacentric, then acrocentric and acrocentric, and the fourth one is telocentric. Right? Metacentric means the centromere is almost located at the center, right? That is with almost equal arms. So this I am drawing the metacentric chromosome. Right? With the primary construction, secondary construction, and the kinetochore proteins. So metacentric, almost equal arms are there. Within the submetacentric there is unequal arms. So equal arms, almost equal arms, unequal arms, and because of the unequal arm, the one arm is small and the another is large. Smaller arm is basically a P arm and the big is called as a Q arm, like this. So this is a smaller arm and this is a bigger arm. This is a P arm and this is a Q arm. In acrocentric, very small and very large arms are present. So this is 1, this is 2, the third I am drawing here. That means they have subterminal centromere. That means centromere is localized at one arm only, in such a manner that one arm is very short and the another arm is very, very long. This is the acrocentric. And the telocentric is basically terminal centromere. This is subterminal, and here it is terminal centromere with only one arm present. Right? So with this we complete the structure of the nucleus, nucleoplasm, chromatid, nucleolus, nucleosome, and the chromosome morphology. So this chapter, that is chapter number 8, is over, and the next session we will start with the cell cycle and cell division.